I was gonna cover,
Got kicked.
yeah, okay. It's possible that my connection, I might drop out back again. I'm calling in from Malawi today. I'm here for probably 5 more days. Um, and one thread that I'll do is I'll talk a little bit about why I'm here, but there is a big diagnostics meeting of sort of African manufacturers starting to do manufacturing of diagnostics in Africa. So I had to come out for this meeting, and I'll share a little bit of what I've learned so far. But then I want to cover some threads on kind of big picture challenges on the oceanography side, especially because a few of you have mentioned interest in microplastics. So I'll just cover some of the basics of case study of some tools that we have built in microplastics. And we're going to then transition and also talk about any of the Fred parks that might be there that are for microplastics as well. So that's sort of the agenda for today. And before I start, I just want to confirm, show of hands if you don't have a team as yet. So raise your hands if you don't as yet have a project team between 2 to 3 people. Uh, I'm not seeing any hands. I see
There's
a
two in
hand.
the classroom.
Okay, so there's 2 people in the classroom. Anybody else? Everybody online has a team? Yes? Are you guys excited about your teams? I hope so. That's the whole point of trying to actually do something. Oh, Webber, you have your hand up. Is that a thumbs up or a question?
Um, I do not have like 2 to 3 members in the team, but
Okay, so we should recruit you. You have a project for sure. So because I think we should continue the Nigeria thread. So maybe we will pitch in. Uh, Maybe let's start with the folks who don't have a team. Do you want to quickly choose and describe your Fred Park for a second? In the act of trying to recruit someone that's already in. Otherwise, what we will do is we will sit down and cover a few sets of topics to confirm. So, anybody in the uh, kind of on the Stanford side, I saw 2 hands. Do you guys want to just briefly describe the Fred Park that you did or the problem statement?
Yeah, I can share really quickly. I
Yeah,
did something about like microplastics in oceans, so we can also talk about this later as well. But I think my interest
yeah,
is like more across the ocean. So I've talked to a couple other people, and so I'm not in like a rush to figure out a team right now. But yeah, I'm generally interested in like
yeah. No, no,
microplastics.
no. No, you have to be in a rush to figure out a team because we're going to run out of time. I'll cover primarily a broad sort of basis on the ocean side of the story. So I think at least as long as that problem space is there, then it's okay. And let's go around and we'll see whether anybody else is actually also interested in engaging on the microplastic side of the story. But let's do that after the class. Okay, but at least you have an interest space that you're deeply excited about. Um, okay, I saw another hand, and then I also see Camille's hand is up as well. So let's go to the Stanford classroom. Did somebody say they also don't have a team as yet? And if that's the case, can you describe your Fred Park briefly?
Interested in doing something more on the global health side, like infections, and lower resource countries. And you said
Have you
that
found and looked through the teams to see uh, anybody doing diagnostics? Isn't there any diagnostic projects yet? Anybody else who's on the call that is leading a diagnostics project? Or what did you write your Fred Park on?
infections, like skin infections.
Oh, skin. I see. Yeah. I think that's an interesting thread, whether we revive the older thread because so much groundwork was already done. And it actually is a very tangible uh, and when you start thinking about, I was looking at the UV index of several countries and realizing that for all kinds of reasons, there is a very direct path of thinking about also skin cancers associated with it. Okay, so I think we will talk about it after the class. Let's cover that. Camille, I see your hand is up.
Hey, yeah. Hey everybody. I think maybe it's something you maybe wanted to say for later, because what I was just
Mhm.
gonna follow it to what Luke was talking about. He and I actually met and he met with several other people, so I'm definitely involved in that. And I didn't put anything on the Fred Park myself because what I did is
Mhm.
I went through and I actually put some notes on folks. So anybody whose notes I put that I'm definitely interested. And there was someone who had something about like potentially doing some type of sequencing, in the field. Anybody who does sequencing knows that the mid ion is not it.
Mhm. Mhm.
So if there's a way we could test for genetic information out there, I'm definitely interested. So this is also kind of like a public announcement for whoever wrote that. You know, put it in the
Yeah,
chat
so
or something, and I'll reach out.
yeah, I think unless I see, I will, after this class, I will go through and see if there is any broader oceans teams and folks that are thinking about problems in the ocean can just band together. We can cover, we can tie in techniques and puzzles all together. I think at the end of the day, it would be valuable to be aligned around a specific class of projects. So I think the big request that I have, or it's kind of a stronger request, it's not an option. Everybody should now have a team other than the two, three people that just spoke. So please make sure that you are meeting with your teams regularly. And one of the key aspects of this is that, I mean, I was looking, we have less than a month left. We will have presentations on the 11th of March, which is the last week of class. So I think working backwards from there, it makes a lot of sense. Now we will transition very much into this one-on-one mode. This is really where rubber meets the road, where we can really spend time talking about ideas, immediate sets of things, certain sets of scenarios of what do we want to do to prove these ideas have legs or not. And so I spoke to Alex and Enrico. They're going to open up a standing office hour at this point. And simultaneously, I will open up an one-on-one office hour, essentially starting instead of lectures, I'll just sit down with every team for half an hour. And so we can choose a date. I think I was thinking about 25th. Uh, but the important thing about this is whenever you're sitting down with me, sitting down with Enrico, sitting down with Alex, make sure you're really prepared with all the sets of things that are going through your head and you can organize them in a certain way. Because one important thing we're going to try to do is in the design analysis side of the story, as you start following through your Fred Park, you will have to make choices because you cannot follow through with all the ideas that you're thinking about. And it's perfectly okay that you choose one of the ideas or sort of a solution type and then work backwards from there and hope that that's the one that has the most legs And it might turn out that you are able to prove by the end of the class that that was the wrong way to think about it. And that's actually also fair. That's good. But it's very valuable that you all meet as a team outside the class time so that you're starting to get to know each other. I'm seeing a few hands. I'll send a message to Shima to check what's happening with the shipping, honey. So we
Yeah,
will get back on
I can
that.
update
Yeah,
on this thread. We
yeah,
received we ordered some envelopes. So we'll be sending I think 5 full scopes per envelope to everyone, and then we'll collectively send a larger shipment to the Ashoka cohort as well.
Ashoka. Yeah,
Yeah.
yeah.
So
Can
that should
Enrico,
be
can you
sent
just
out
make
this week.
sure? Yeah, tell Shama to ship today if she can. It
Yeah.
would be really nice to kind of get this out now. Um, and I see Kennedy has a note about poultry diagnostics, and I think we'll get back to it. Okay, all right, let's switch topics for a little bit. I think it's ironic that we're doing this a little bit late in the class, but I want to start with uh, something on the front on the threads around frugal oceanography tools. This is something that Enrico directly works in itself as well. But I want to share a class of challenges, and I think I'll share them uh, slightly. I'll start with a very sort of a personal note and how I've been thinking about recently a class of problems. Then I'll end up generalizing it a little bit so you get to see the sense of what's been happening. And then I'll talk about one or two tools that we've been building in the lab in this space. But this space is quite large. Figuratively and literally, because the ocean is the largest biosphere on the planet. And quick show of hands if you've actually spent time on a boat before. How many of you have? I see Serena for sure. I see some hands in the class, which I am actually surprised. So I know both Enrico and Alex have been out on boats. And I am quite surprised that there is a lot more hands. And so I think, one of the important things to understand is it's quite hard to think about challenges in the ocean if you never experience the ocean. And, one of the things that ends up happening is there was a framework for the longest time where we believed that the ocean is an infinite resource and we could just do whatever the hell we want. And that turned out to be a big mistake. And so I think many a times policies that were built and set up did not account for the fact that we can literally fish the ocean. We can completely fish out the entire biomass in the ocean if we wanted to. We have the technologies now that we can play extremely destructive approaches. And slowly that has changed and dawned on people. Um, and I think one of the things that's valuable is I'll talk a little bit about the history of oceanography and that as a field and how lacking are the measurements to begin with. And this is sort of at least the big question that I want everybody to think about here is how do you go about measuring uh, the ocean? So let's take a few seconds and tell me in just ideas that pop out into your head. How many of you have heard this frame that people have said that, oh, we know more about the surface of the moon than the bottom of our ocean? I'm assuming everybody's heard that before. 70% of the planet is the ocean. And if you were to think about what type of an approach would you take if you had to go and make measurements out in the ocean. You can talk about current techniques that you might have heard of how measurements are made in the ocean, or you could actually dream something that you're thinking in terms about. I'm just curious, when you look at the moon, it's pretty straightforward. Either we send a probe to the moon or you point a telescope to the moon. That's pretty much it. Sometimes you can actually shoot lasers at the moon surface, get the reflection back. From that, you can do a lot of chemistry. You can figure out what chemistry is on the surface of the moon. So I'm curious, just let's brainstorm a little bit. How do we go and measure? Uh, how do we go about measuring what's happening in our oceans? This is sort of rapid fire. So anybody, don't raise your hand, just shoot and shout out whatever you think are ideas around how do we go about measuring our oceans.
A big one is sound. Like we use basically sonar or things like that to pick up
Hmm.
things that are in the ocean, whether it's ocean floor or large schools of fish, basically.
Yeah. So sound is something remarkable because sound travels very well. And in fact, for every one of these techniques, there is uh, this kind of a strange thing that happened that, of course, military is where much of that was first developed. And then it gets extensively used in many of these large fishing vessels. And because we have capacity to measure, they're also able to track many of the schools of fish, which has led to overfishing because they're able to target. And so kind of tools of measurements are dual, they have dual purpose. So that's sound is one. What else? Yeah, Camille, go ahead.
Yeah, I was just gonna say I mean, this is cool. But when we're talking about like, how we measure the ocean, right? I'm more
Hmm.
of the scientist type of like giving communities the access and the opportunity to do it themselves because they have their own, needs and
Hmm.
interests. And I find that most times when you talk with community members, they know what's happening in their spaces. My grandma has
Hmm.
grown up in Philly for her whole life. She doesn't have a college degree, but she understands the
Hmm.
ways that the climate is changing in ways that scientists
Hmm.
have predicted. So I try not
Hmm.
necessarily to focus on like the methods per se, but
Hmm.
giving the methods that are needed and the tools that community members need to actually access
Hmm.
this and understand these spaces themselves. Because
Hmm.
I think,
Yeah, so I
we
think what
talk
what
about the
you're
ocean.
talking about is what you're talking is step two in the process. I'm going to get there eventually once we cover what all tools we have in our arsenal, uh, to then thinking about the purpose of frugal science being that no matter how many tools we have, we won't have the communities and the people, and then that becomes the implementation side of the story. So let's just first go through. I just want to get a sense for the kinds of technologies people have been exposed to, and then we will come back to this point, Camille.
Mhm.
Because the case study that I'm going to use is exactly around the community side of the story. And then I'll ask you to add in terms of other sets of tools that can be deployed on the community front.
Oh yeah, well there's
Um,
definitely we talked about sonars, but there's also opportunities, where we collect sediment, we collect water, and then we analyze where for what's actually in there. And then of course there's
mhm,
visual as well.
mhm.
Visualizing
Okay,
what
so
the
let's just make a
spaces.
quick list. There are cameras underwater. You think about using direct footage. Most of you have seen many of the research vessels publicly broadcast their footage. So there is vision and camera. There is, Camille just pointed to biochemical and biogeochemical analysis of the water. So many of the types of diagnostics and other tools that we were thinking about could directly be applied. So that's directly analyzing the water. What else? Just let's keep populating other ideas. Yeah, just shout out. I see a couple hands in the
Um,
classroom, so you can just shout. Yeah.
one thing they do is like tagging animals, like to figure out like
Hmm.
migration patterns and stuff.
Hmm. So tags are a very important piece of the puzzle. And I'll see if I can find that video. I mean, some of you have definitely seen whales being tagged, literally going around the world. Uh, and you can again also think a little bit about the cost side of that story and what percentage of animals should be tagged. What else? Yeah, go ahead.
Like you do like the chemistry in the water, like pH, how much oxygen is dissolved.
Hmm. So you can take any analysis that you can imagine. If you can do it on a water sample, it should be applicable. There are standard probes, oxygen, temperature, chlorophyll. Many of them are done in situ. So what's very powerful is also you can do this not only by collecting water, but you can put your instruments directly and think about making that measurement directly. What else? There is a big one that nobody has mentioned, which I'm surprised. Ian,
Um,
go ahead.
you can use satellite imaging, I think,
Satellite.
to look at the
Yes.
ocean from above and
Hmm.
look at more global behaviors or currents.
Hmm. Yeah, so I think satellites completely transformed oceanography. And one of the famous ways that it occurred was often enough when satellites were being launched, it was always about looking into space, then it turned inwards. And the most, I mean, in terms of just thinking about over the last 30, 40 years, just remarkable improvements have been made in the kind of satellite data that comes out. There is several of these hyperspectral satellites have been launched. People are trying to extract as much information they can extract from far away. And if you do from cost analysis point of view, it's very interesting that satellites fall into a frugal science category because although it's a single launch, but if the satellite is going to be operational for 10, 15 years, you divide up per data point, you can actually get quite a lot of information from some of these larger maps. Um, any other sort of more ideas that you can imagine? What else comes to mind in terms of thinking about measurements in the ocean? So clearly, plastics and pollution, oil spills is a big one. You know, it's not a standardized measurement that's out there, but clearly oil spills don't just happen on the surface. They happen volumetric. Rigs have a requirement that they need to go check. Some of the infrastructure is actually checked on a regular basis. So there are certain class of measurements that are made along that way. Uh, there
You
are
can
community
also
divers. Yeah, go ahead.
you can also check seismic activity and
Mhm.
extrapolate things about the ocean floor based on that.
Yeah, so that's a very interesting one. I was going to introduce that paper a little bit later. Uh, there is a recent interest right now, and only the, I would say the tip of the iceberg only has come out, which is most of you know that the only reason I can talk to all of you and you all can hear me and our friends across the globe is because of these optical fibers down in the ocean right now. So there is a massive bandwidth of optical fibers that go through the ocean. And almost a decade ago, people started thinking about what if you could infer something from that about the ocean. And so the first thing that people have been able to do is test for seismic activity. But there is a lot of broad interest. It's not so obvious how you would go about doing that, but there is an incredible bandwidth in the cables that have already been laid out. And I would say it's one of the most underappreciated infrastructure that already exists because they have repeaters. every few, I think I don't know, Enrico, if you remember it, what is the fallout ratio in a fiber? My intuition was that every 30, 40 kilometers, you definitely need a repeater.
Yeah, an amplifier. Yeah,
Even,
intuition as well.
right? So then suddenly you realize that there is an incredible amount of electronics and tools that are put on this cable that are then anchored and layered at the bottom of the ocean. Currently, they're not making any measurements about the ocean, but they could. And so I think this is kind of an untapped infrastructure that comes into play. Uh, and then I think people didn't bring that up, but of course, plastic pollution is a big one. There are NGOs that go out there, they do surface measurements to just capture and measure total plastic that's out there. There are all these removal efforts associated with that too. Uh, but there's a fundamental challenge in everything that we have thought about in the context of the ocean. And when you do the numbers, and if one of you has done sort of by the numbers on this or a Fred Park on this, the total volume of the ocean, if you take 70% of the planet's surface and 5 kilometers, 4 kilometers average depth, it's the largest biosphere on the planet. So one of our challenges per se here is that no matter what approach you take, you're talking about one of the largest ecosystem or a sub-ecosystem on the planet. And that makes it very difficult to be able to get comprehensive view of what's actually happening in the ocean. So this is sort of something that is hard to fathom if you were to think about this, that, 70%, the largest livable space on the planet is the ocean. And then you could argue that it is also the largest unknown on the planet is the ocean, because by volume, if you have such a large biosphere where life exists, it's also the largest unknown.
He'll reconnect.
It did look like his computer battery was really low.
Aren't you doing ocean stuff?
Yeah, a little bit. I can share some stats as well. So I had like a group meeting presentation. So I mean, I'll just while he's connecting, I think it's interesting. Like actually, the ocean is the largest carbon sink. So it actually absorbs like 30%. Oh, I think. Oh yeah, my bad.
Okay.
Yeah,
Can you
yeah.
guys hear me?
Okay.
Yeah, Enrico, my computer suddenly died, and I am trying to turn it on. So actually, while I make an effort to turn it on, okay, it's turning on now. So we'll continue a little bit, and then Enrico, at some point of time, I might ask you to share slides if I can't share slides through here,
Sure.
especially on the set of measurement tools. But I think maybe the punchline that I want to mention is up till now, we mentioned the threads associated with sets of tools and how large the system is. But one very critical thing to also just remember is 1,000 different ways in which the ocean influences our life. And kind of one of the big ones that I want to spend time talking about is carbon sequestration. So I'm going to try to see if I can connect back online. Actually, Enrico, can you share your deck for a second? Because it's going to
Sure.
take me a few minutes to I have to reconnect to the network somehow. And let's see if I am able to reconnect again.
This slide on carbon relevance. Let me try to share it.
Yeah, just open the total carbon sequestration slide for a second. And my hope is that I should be yeah, so okay, I think I was able to get back in. So
Okay.
let me hang up here. Uh let's try okay, sorry about this glitch. Let's see again. Okay. So I think the way that I want you to visually think about the ocean is a little uh, comment that we were just talking about is often enough one of the challenges with satellites and in general the approach that's been taken in trying to think about the ocean has really been we've been taking a surface picture. And so this is a painting that artist in my lab, Rebecca, she painted this because we've been thinking about this idea that if you could cut somehow a chasm into the ocean, that's when you would deeply appreciate the context of depth. So every single time you look at a photograph or a satellite picture, what you're looking at is a tiny, thin skin of the ocean. And it's given us this false perception that we are actually observing the whole ocean. And in the bulk of the ocean is really both the remarkable life and the chemistry that is at play that is critical for maintaining most of I would say, the homeostasis on our planet. Every aspect of what you think that the planet maintains a homeostasis around, the ocean either play the most dominant role or a very important role, whether it be temperature, whether it be precipitation, whether it be the total amount of wind energy along the way, whether it's the amount of carbon that's sequestered out of the CO2 of the atmosphere, so hence the total amount of carbon in the atmosphere. And one of the challenges that has really been there is we don't have any way or approach to be thinking about a comprehensive picture of measuring the ocean. And then this is an old slide, but it's an important number. And I don't know if you all can actually see
No,
this
Manu,
as a video. This is supposed to be a video. Ah, that's too bad. Actually, you can't see the video play out. This is from a group at MIT. I really like this. Many of you can read about it. It's called the MIT Darwin Project. And they did some simulations, and very similar to what you're used to seeing in terms of trees grow in the summer and disappear. Very similar to that, life appears and disappears in the ocean in a very dynamic way. And so the color here represents living plankton and living plankton of different kind, and we'll kind of get back to these plankton in a second. But I think this is when I learned this, I would say maybe 10 years ago, it's kind of ironic. I grew up in India, never saw the ocean ever. And I don't know how many of folks, uh, I'm hoping all of you have at least visited the ocean once. I only saw my ocean for the first time in Boston. And then when it's out of sight, it's out of mind. And one of the shocking realization for me was just this idea of 93% or so of all heat that we generate is being absorbed. And this number is debatable, but I would say between 30 to 40% of every CO2 molecule that we emit is also technically absorbed by the ocean. Now, how that happens is something that a lot of us think about, and we'll talk quite a lot about that. But there's a class of estimates that are associated that if that pump or the mechanism by which the ocean is actually absorbing the CO2, if somehow that was to switch off, you would go to whatever. What is the current number, by the way? Does anybody remember? What's the current CO2 number? I used to have that tracker on my phone all the time. I think it's definitely, if somebody Googles that number, we've crossed, clearly it's 415 or no, 420. It's something that you guys should keep a track of.
420.
Uh, Serena, what's the number?
I think it's over 420 ppm.
Yeah, so 420. And then you can, whichever year you were born in, you can use that as a proxy year to see how much it has changed even in your lifetime. And it's a very relevant way of thinking about that even in our short lifetimes, there are these dramatic changes. And one of the big questions around, uh, which is quite perplexing, is neither do we understand deeply the mechanisms associated with sequestration. We also don't understand the tipping point of many of these ideas. So a tipping point being is it possible that at some point of time we might actually lose this capacity to sequester the carbon? So I'm just trying to tell you that the ocean are incredibly important in many ways and forms. Uh, I think there is a video that I want to show you guys. I was in Chile recently. We have a new program that we've been thinking about in terms of HABs. And this is a photograph from 2021. And some people here in this call works on HABs. And I was shocked to see that this is now these are harmful blooms. These are algae that would appear in the ocean, seemingly as if they appear out of nowhere. It turns out that they are resident in the bottom of the ocean, and then they can come and they generate massive toxic blooms. And the picture that I want to show you, which I found shocking, which is, with this, you sort of see what's actually happening. And in Chile, I'll see whether I can pull out that number. Over the last, let's look at this number. This is a number from 1988. You can see this is 1960. And this is the count of number of HAB events that occur in Patagonia. And so starting 1990, exactly when aquaculture is taken off, there's this skyrocketing of it's a very, you can say that we don't understand when they occur, but it's very obvious. We have fundamentally changed something. And then you might say, why do we care about these HABs? Yes, you can't eat the fish, but this is what I find quite shocking, actually. So this is an event that occurred in 2015. It's the largest mortality of sei whales that have ever been observed. So 343 whales died right when the bloom happened. And it's actually hard to prove that such a large animal is dying because of toxic blooms. And some of those stories are just coming out. It's taken more than 10 years for people to find these toxins into the body of these whales. So it's not published yet, but I recently met the group that's been working on this for 10 years. It's quite a difficult problem because the moment you find a dead animal on a beach or a mammal, you realize that it's suddenly decaying. So any measurements that have to be made immediately. But I think one of the key factors around this was associated with the fact that there is a massive amount of aquaculture that happens in Patagonia, lots of antibiotics, very similar to what Kennedy was talking about in the use of antibiotics and other uh, chemicals in poultry. You can think about a similar thing playing out in aquaculture. And that has an incredible detrimental effect. And I think this is the most depressing video, but I need to show this to you guys. This is from a friend of mine who's a diver in Chile. And I'll just let this play out. And you see all this pristine biodiversity, but every single thing that he is touching is actually dead. So this is sort of mass mortality events that occur in the ocean. And unless he was there poking at it, I mean, literally every single one of them is dead. So they are just holding their shape. Uh, and ironically, in many places, if something like this would happen on land, somebody would notice. But on the other hand, because many of these things happen underwater, there's also wasting diseases. There are new viruses that are popping up. And I mean, the big thing that I'm trying to convey here is unless we increase our capacity to observe, we will be losing these ecosystems even before we have found them. And that kind of is really true for the ocean because we do not have ways of being able to see what's unfolding in our water columns. And, I think now it comes to be thinking about the kind of approaches at play. But these sets of approaches, we can debate about what are the best approaches. There are ways of thinking about completely autonomous approaches. There are ways of thinking about community-based approaches. But one of the biggest fundamental challenge that we face in oceanography today is that the cost per data point is way too high. So you end up paying, when some of us go out to sea, we pay between 50 to $60,000 per day to be at the open ocean. So that by itself is quite expensive. And then you can ask, like, why is that so? Could we build a class of robotic platforms associated with measurements? And I think, Enrico, I'll unshare this. If you can share a slide from your side. Actually, maybe I'll show the Argo for a second, and then
Yeah.
you can do that comparison slide. Uh,
Okay.
so, people have been thinking about a class of automated monitoring platforms, and then I'll switch to talking about the community monitoring platforms. Uh, but this is a very interesting idea that kind of got started, has grown, and so what you're looking at here is the map. It's not the most up-to-date map, but it's a decent map of what are called Argo floats. It's a device that looks like this, has a really cool buoyancy engine, so it can tune, and you throw it. It has some telemetry. It has some physical sensors, and now the most recent version of them actually also have biological sensors, including chlorophyll and a couple other factors. But you should think of it as a platform, and they report data for a couple years, and then the battery dies out, and they sink to the bottom of the ocean. So I would say, it's one of the most powerful monitoring network in the bulk water. And any of you could go to this website and you can pull out data from the Argo floats. And the reason I'm bringing this up in the context of frugal science is I was quite surprised when I pulled out the cost estimate for one of the Argo floats. This is in $1,000, and you can see roughly deploying one single Argo float is around $100,000. So now you multiply 100,000 by another 1,000. This is too many thousands for me to count. And you start realizing that this is an incredible, I mean, of course, it's a remarkable infrastructure, but it's not sustainable in many ways the way we think about. And again, they also have a certain amount of lifetime. There's no fundamental reason. There is lots of technologies have transformed. And then you can start imagining very much what is happening in space technology. Space is only accessible because people figured out how to do operations at a much cheaper scale. And ironically, that same thing has not happened completely in the field of oceanography. And that's why it's important for some of you to pick up a class of ideas and apply these thinking into measurements in the ocean. I'll give another example, and then I'm going to have Enrico share a class of tools. This is an example that I love. This is from Alistair Hardy, another oceanographer. And what you're looking at is a citizen science. I mean, this was not a citizen science. It could have been done in a citizen's way. But he invented this really clever idea. These are called plankton recorders. It has it's something that you tie to a back of a shipping vessel. Because literally, if you look at the shipping traffic, I don't have that traffic picture here, but 15 to 20,000 shipping vessels. I mean, most of you have seen that in the bay, all these ships coming back and forth. Uh, so what this device does, it's a totally passive device that gets put behind the ship, and you can do biological measurements. It's limited in many ways, but at that same time, it's actually very valuable that one single person's invention led to what now is one of the largest data set of its kind, because these transects keep happening. This object has no batteries, nothing.
Okay, just while he's reconnecting, I'll just share the slides he was
Yeah,
mentioning.
Enrico, yeah, I'll let you share, can you share the class of measurement
Yeah.
tools that you had put the deck for the group meeting? And then
Sure.
I'll come back and I'll close by talking about planktoscope in the last 5 minutes.
Okay. I hope
Yeah.
everyone can see this. Yeah, this slide was meant to argue that there's like a significant gap between macro surveys, like we were talking about, which are like sonar mapping or satellite imaging, and lab analysis, which is like yeah, the biochemical side of things. And the way that like that gap exists in measuring, like, let's say mesoscale events. And this is typically done by
Enrico, I don't see your slide. Do other people see the slide?
Yeah, I see it.
Yes? Okay, all right. I don't see it, but it doesn't matter. Continue. Yeah.
and and so what's really cool about these Argo floats is that they're doing a trisecting the entire water column by moving up and down, and they do this by changing their buoyancy, and they're actually very energy efficient because just by changing your volume to 100 milliliters, you can actually move up and down almost 500 meters. And that's because you need to match the pycnocline, which is the density gradient of salt water, which is actually doesn't change that much over those depths. And that does primarily vertical mapping, and then you passively drift in the ocean. But there's another technique called the gliders, which still uses this phenomenon of buoyancy, but you actually introduce wings and tails. And by having small rudders on your rudder or small servos on your rudders, you can actually horizontally map the ocean as well. So you can program your robot to, travel like kilometers of distances in the horizontal direction by still using this buoyancy phenomena. And here are some other imaging in situ tools. So for example, here we have a lot of plankton recorders. So these are all imaging devices that you would attach on gliders or other platforms. This is actually measuring just particle size. So it has just a laser and a detector, and measuring by how much your laser is scattered, you can estimate the particle size. Then you actually have cameras, and then you have some more other techniques like fluorescence microscopy you can start trying. And this is actually a holographic camera. So here you get 3D reconstruction of your like image field. Yeah, that's yeah.
Yeah,
I don't know if this is worth
if you
mentioning as well.
unshare, I'll share my side of the story for a second.
Okay.
Yeah. So I think kind of one of the things around this is just on the imaging front itself, one way to map what's there is just this is the class of tools that have been developed. And we had been looking at these tools for a while, and we use many of these sets of tools. You can say they have different names. On the very left end is actually viruses, the smallest possible thing that you could measure in the ocean. And on the very right are, jellyfish and other larger things. But ironically, when we started thinking about most of these spaces, every one of these instruments used to cost between $70 to $100,000. And very much in the spirit of frugal science, we decided to change that. And that led to the first version of this tool, which is now called Planktoscope, and now it's commercially available. Is actually this was the very first instrument that we built in that scenario. And I am assuming, have people seen the Planktoscope yet or not yet? Enrico, have we done a demo for that? Some
No,
of you
we have
have
not.
them. Okay. Yeah, so maybe Webhub, you're going to be running a Friday demo because my unit is not with me. My unit is in Peru right now, and Enrico doesn't have one of the units. So, but you have a unit. But we'll talk about it. But that led to this project, and kind of one of the primary goals that we thought about at that time for this project was, could we enable sailors and citizens to make these class of measurements? And I'll talk a little bit about this as an instrument. This was the first version of what we built. And then eventually it's evolved to become the object that you will see. This is the actual evolution. So it's kind of fun to watch. This was the first version. This is Thibaut and Hong Chuan, were the first people in the lab building them. Then Thibaut built the second version of this, which we over-designed certain things, and this was very fragile. It never used to work. And then finally, what ended up becoming the stable version is actually this. No, this is another version, and now the stable version is this. So I'll just walk through, and Weber will show you all this instrument in a little bit. Now, for around, I would say, $1,000 in parts with a Raspberry Pi, an imaging camera, a simple pump, we now have the capacity that you can take water either in situ or water that you've collected, and you can do flow-through imaging on this system. We released this completely open source. And so just to give you an idea, this is the type of data sets that come out of it. So what you're seeing is things just passing by and you're not even able to see because there is a glass capillary in the middle and the samples are just passing by and the camera is just taking pictures continuously. And from this, we can build an entire statistics associated with what you just saw. So I can take that data and I can convert it into every segmented object, and you can analyze this in ways and forms. Then we run all kinds of machine learning to be able to identify species. And then we are able to build these, what we call ecograms. And these ecograms are associated with now biological diversity at a given place at a moment of time. So that's kind of the framework of going from what existed before. And then we were asking ourselves, how do we share it? And one of the key things that we did at that time is we just publicly shared every detail. So any of you can go to this site. It's called planktoscope.org. And what I was surprised by is I started getting these photographs from people around the world, and they started building it. These are people that we don't know. So any one of you can just take the instructions, go on Amazon, buy parts together. We started realizing that we have to support certain class of communities that led to the community programs in similar spirit to what Camelia was talking about. And that's the current deployment of the tool. There's, I would say, we crossed around 200 plus units are now being used. And anybody has access, and this is quite an old map. So I know we have lots of units in Africa and lots of unit in Latin America now through the sets of programs. But one of the perspectives about this as a tool is that once we recognize the gap, I would say technically it was fairly easy to fill that gap once we understood. And the direction that this is going, which is there is a couple of NGOs now that are pursuing an idea of taking sailors. So this is, for example, all blue water sailing boats, between 10 to 15,000 sailboats currently are on the water as we speak. These are citizens and people who just love the ocean and love to spend time in the ocean. And so one of the key programs we've been building is taking these sailors and training them and installing these on their boats. So this is sort of one of the programs that Plankton Planet ran. And I like the joke here is they're not citizen scientists, but they're citizen scientists. Uh, in that is also a genomics program as well. So they're imaging and they're using their cooker to actually dry out samples. And then these dry samples are shipped for genomics. But you can see these filter cakes being prepared. And one of the things that we were able to show in this recent paper is that the data sets collected by these citizens actually matched in quality the kind of data that has been collected by Tara. The frequency is not that often because these sailors don't like stopping for sampling. If they catch wind, they want to go. And so that led to a design of a new kind of a net that you can still throw. So you can see these are the plankton nets. I mean, eventually we need to redesign these nets because even these nets can cost $1,000, which doesn't make any sense. But we actually designed a new class of nets that are high-speed nets where you can throw a plankton net and not have to slow down. But one of the perspectives of this as a program is that very much like Full Scope, it grew organically, and there are many variants of it. And now there's a couple of variants that we have tried in the lab. The variant that Webhub will share on Friday is we're actually using now the same tool for diagnostics in urine, very much like the ocean when you're looking for single cells. Urine can actually be used to diagnose many sets of conditions. So I was thinking about this for a while, and then finally I was able to convince Webhub, and then he went out to Nigeria, and now it's a full circle. We didn't design this tool for human diagnostics, but we've now realized there is a huge value because it allows us to image large volumes rather than smaller volumes. Then there is a configuration that you can change in this to actually redesign it for microplastics. So that would be a fantastic thing. A lot of collaborators have been asking. And we have done one module in which we can incorporate fluorescence. And fluorescence is a classic first step in thinking about microplastics. The second layer to this would be is we've also implemented a portable Raman, and then you could also identify what exact plastic was there. And so I mean, the reason I'm mentioning many of these modifications is then you don't have to build from scratch, but you're adding a new capability to a tool that already exists that has a very wide adoption in terms of community. And it has a capacity to then build functionalities that anybody else can replicate. So one of the things that we're going to try to do is Friday do a quick set of demos. And I think Webhub, you'll have to run that demo. And then you can try a pond sample or you can just get a water sample.
I have a quick
Yeah,
question. So when I do the
yeah.
imaging on the planktoscope, I will not be able to connect the computer to the Wi-Fi. So should I emit some
Y-
samples beforehand to?
Yeah, so I can tell you a trick. There's a couple of tricks to put. What you do is you connect your computer to there is a trick that we had in which we can zoom in and do a remote. Yeah, I'll send you a script for it. We have a trick to be able to actually connect uh, Planktoscope as one of the Zoom participants. and
All
then we
right.
just share the screen from there. Yeah. So, but
Good.
I have to remember that trick, but I'll tell you that trick. Yeah. That way you can do a live demo associated with it. Yeah. And I think the other option would be is somebody at Ashoka is holding a camera while you're doing the demo. Um, so I think, yeah, that's probably, I know some of you have to run. This is a tip of the iceberg, quite literally, on kind of the framework of oceanography. One of the things that I would say is there is a tremendous number of tools that have been built, but none of them have ever been built in the context of thinking about scaling and the kind of cost. And one of the important hypotheses around really here is that as you start growing the kind of measurements, as you pass these measurements into hands of communities, you have this level of scale that was never possible before. And you know, in the context of the ocean and what is being described as the blue economy, with I mean, I was doing this calculation and I found it surprising that although the ocean is the largest biosphere, less than 1% of our proteins actually come from the ocean. And even at that 1%, we are destroying most of the ecosystems. So clearly, we don't know how to do this. But again, if you realize the number of people that live on the ocean, and for them, it's 70% or more of their protein needs come from the ocean. So these numbers are quite staggering. And one of the threads, another one that I really like is between 90 to 95% of all plastic that we are dumping is actually missing. We can't find it. So it's the dark matter of plastic. It is, we believe it's in the ocean, but if you just look at the plastic on the surface, that does not account for 90 to 95% of all plastic. So it suddenly, you start asking like, oh, is it possible that much of it is going in ecosystems that we are not measuring, which is the midwater and the deep water? Because many of these pollutants are actually reaching deep water now. And this is sort of, again, goes back to that none of these issues can be tackled unless we directly tackle the measurement problem. And that's why we're focusing on measurement per se. Okay, so I'm going to stop at this point. I know I'm already over 3 minutes. We'll turn this into open mic. Any questions on this threads? Questions, ideas, especially the folks that were thinking about doing a project on the side of the ocean. Does something spark your interest? Oh, by the way, something that I said, I mostly use the word ocean and I talked about the ocean, but in general, I really mean water, because fresh water is going to be the most precious resource in the next 5 to 10 years. And just like we don't measure anything in the ocean, we actually also don't measure our fresh waters. So every idea that you're thinking about actually directly applies to freshwater itself. Chile, for example, has 1/5 of all of the world's fresh water, and all the glaciers are accelerated and they're melting much faster. So you end up thinking about, I mean, many of you know that UP will run out of water pretty soon because the Himalayan glaciers that provide much of the drinking water will not have water. And so the same framework that I mentioned about marine ecosystems actually also applies to freshwater. And I don't know how many of you have followed the Clean Ganga initiative. And, you know, I want to see the data. You know, when people say that, oh, we're going to clean this water body, where is the data? And much of that is not implemented because many of these things, you could automate much of this to have dashboards of data sets coming out from places. But much of that is not done. And, you know, it's another layer, which is a much harder challenge, but really worth thinking about is the bacterial side of the story, which we discussed a couple of weeks ago. It's like, oh, now you don't want to just know the species that you can see, which is 10 micron and above, but you want micron. You want to know viruses. And this is, again, sequencing kind of crosses that path in very interesting ways. Yeah, Ayan is saying he lives close to Ganges, and uh, there's all this thing where they say they sell the pure water of Ganga. It would actually be really valuable to put that in a planktoscope and see, uh, collect a data set, because in India now it's sold. You can buy the water from the purest river and it's not so pure. Okay, comments, questions, ideas, open mic.
Yeah, I was curious as to like first of all, like you mentioned that going out in a boat or going out to do the research costs 50,000 a day, and
Mhm.
the Argo tools are like
Is
100,000 per.
100,000.
What's like the biggest cost? Like what's the biggest cost in that?
What do you mean in the Argo float?
In both of those, yeah.
Yeah, the boats, I mean, the boats are guzzlers of diesel. You have to have an entire operation. You have to keep people safe. Efficiency of, I have a friend who's actually designing a solar powered boat right now. Not so much for research, but it's not totally crazy to think about that you can build much more efficient boats. But then the infrastructure costs would be very high. Those boats would cost quite a lot. So even if they reduce their cost, much of that cost also goes in salaries of people and other things. So what you can do in an autonomous instrument, you should always do in an autonomous instrument. There will always be a need for scientists to be in the ocean because we are thinking on our feet. We're trying to do and plan a lot of that. Uh, and then in the Argo side of the story, I don't really see any reason why they cost $100,000. If we put our mind to it, you can technically definitely reduce that cost down. They are not recycled, which I find shocking, because currently the cost of picking them up is higher than the cost of making them. So if somebody's Argo float is floating on the water, you know where it is. It has a GPS coordinate, but you can't go pick it up because it'll cost more for you to pick it up. So then imagine kind of what balloons do. It's like, oh, what if it had very simple algorithms that it could utilize the currents to show up in a given region and then it could be picked up.
Yeah.
But again, I mean, much of that infrastructure that sinks to the bottom of the ocean.
Yeah.
Yeah, I think Enrico in his last year's class built a low-cost buoy, and then we've been designing a low-cost buoyancy engine. So there's lots of pieces that are there. And I think one thread is thinking about the chassis is not that complicated. I think it's the sensors that you add. It's the chassis is around 20,000, but then what you add to it is really adds up the cost. Yeah, go ahead. I see Honey's hand up, and then there's a question in the class as well. Yeah,
Yeah,
either
I
one. Yeah,
was
Honey, you can go ahead. Yeah.
yeah, I was curious to know that is there any real-time monitoring system to monitor the cleanliness of rivers? Because many of the times we do it not longitudinal wise, whether we do a
Yeah.
test and then, there is no continuous
Yeah.
data or information flow uh, to the
Yeah.
most populous or polluted regions in
Yeah.
India, or we
Yeah.
talk about many.
Yeah. So I think we have discussed this many, many times because we are currently building a continuous version for the planktoscope just for monitoring biodiversity. And there is a version of an instrument like that that could be used to just do bacterial counts as well. So that's doable. There is also in parallel, the wastewater sequencing tools have now come online. And I've seen them used for many respiratory viruses and others. And I think there, the trick is you have to be good at sampling and making sure that you have clean sampling protocols. But it's not a continuous, there is an attempt of doing continuous DNA extraction, but clearly, uh, Actually, I do know a talk of underwater sequencers and stuff, but it's just is not the right approach. I think if you don't want to know the identity, but you want to know a microbial load, I think that could be done. And then you could connect that with rainwater, for example. And you could connect that with different sorts of events that happen.
Yeah.
Or you could do something which is very specific. I mean, cholera is the one that I have thought about. If you look at Bangladesh and you look at this massive burst of cholera that comes in and out, that would be phenomenal to measure in real time. And cholera is very doable because just in terms of size, it's I think it's much more on a context of figuring out where to put and then how to build clinical policies on top of it. Like, could you say that if the bacterial count is above a certain number, you could send out a warning to not consume a certain water? River wells and many of the underwater wells also have the same issue because the wells are normally fine, but there are moments of time they're not good. And so thinking a lot more about that type of an infrastructure. And a well is much easier to do. You have power, but a lot of communities depend on it. And right now, there is this idea of a batch test, and the batch test is, once a year. It's not as if people are actually making the test on the fly. Yeah,
Yeah.
I don't know. Did anybody write any Fred Parks on water at all or water access? Because that would be another obvious thing to engage. To me, they are two sides of the same coin, freshwater and
Yeah.
seawater. Yeah.
Didn't do it on water quality, but depending on the water quality parameters you're looking for, like there are some proxies that are more easy to do continuously. Like I think
Hmm.
if there are a lot of continuous water quality systems that look at things like turbidity or nutrients is like semi-continuous and can actually be not super cheap, but
Inferred.
like can inferred, yeah, essentially.
Hmm.
And so if you were interested in that, like something like nutrients would sometimes be an indicator of things
Hmm.
like sewage leaks or like,
Yeah.
that would be correlated with disease or like growth of unwanted
Yeah.
particles.
Yeah. I think the direction I was thinking about was to directly measure microbial biomass. Because
Yeah.
one of the things about this is turbidity is a tricky one with like many glaciers introduce and bring clays to begin with, and they can, but um, I mean, one of the other aspects of this is if you look at the impact of diarrheal diseases, it's such a large impact that you could
Yes.
explore this notion of the that maybe the cost parameter is not so sharp because the gain that you get out of it is so high.
Is
And if it's
so
a more
high.
permanent installation, it is paying for itself or it's a longitudinal kind of a measurement. Uh, I see another hand in the class. Yeah, go ahead.
Yeah, following up really quickly on what you guys are talking about with water
Yeah, yeah.
quality. Something I like to do is I like to surf, and like when I'm out in the ocean, especially after a rain, and I'm like at the beaches, there's always like this huge like flood of sewage going into the ocean, and when I'm surfing out there, it tastes like really terrible. And so, I know the exact problem that you guys are talking about, the water quality, and there's like
Yeah.
on the beaches that say like don't swim, or don't surf, especially after
After
it
a
rains.
rain.
Right, but I
Yeah.
mean, we still do it. Like, no one really cares. But yeah, like I can like taste the difference. Like, it tastes very bad. I'll tell you. But yeah, I'm wondering like one thing is like, can you use the planktoscope, or have you guys like used the planktoscope to kind of like quantify like microbial loads? Maybe you can like have a filter where you try to catch certain microbes and then like have
Yeah,
some.
that's what I was trying to say is that would be phenomenal. There are two things to it. First of all, we can mount much, much higher magnification objectives in the planktoscope. There is a couple of tricks which is called flow focusing. So we can bring and we can convert a planktoscope much more rather than an imaging device, but a kind of a flow cytometer style where you just have a laser beam. And so we haven't directly gone below 5 micron, I would say. But the idea would be is if we try using this, and as long as we are not trying to speciate anything at that scale, if it is just about biomass in the water, that we could do with scattering and just simple scattering techniques. So that would be a fairly straightforward thing to think about. And then you guys should run a back of the envelope calculation to say how much water.
Guess we lost him.
In the class.
I guess just like another idea, I guess, well, unless he comes back soon, is like I wonder if there's also some kind of biochemical like traits, right? Like that could be some like general bacterial marker, right? And so if you, yeah, I don't know, like all living organisms have like RNA or DNA, or like if you like filtered some fraction, which would be pretty easy, like some way to just quickly assay, like, okay, this amount of water had like this amount of like DNA or something in it. And that would, and if you filtered some like small particles, basically you only have the bacteria in the water, and then you said like, all right, we're gonna just see how much DNA, like maybe, I don't know how you detect that, but there's probably a way. But that might give you some assay of like this is the like biomass.
Yeah, I like that idea too. Like we're thinking like right now just using imaging, but like as you
Yeah.
said, like, if there's like other approaches like chemical approaches or like some other
Mhm.
tests you can use to detect. Like something I'm thinking is like whether or not you can attach like primers of like
Yeah.
certain bacterial or other sequences. I mean, this is just focusing on DNA, but you could also do like proteins or any other like I don't know, parts of the cell wall. But like with DNA, you can maybe have like a small primer like target toward a sequence in like bacteria and then like have a fluorescent like tag to that. So like when it binds to its like cognate
Mhm.
sequence, it
Yeah.
should like fluoresce, and then maybe you'll get like a fluorescent detection of like certain
Yeah.
microbes in the I'm
Yeah.
pretty sure that's being done.
Actually, one
Yeah.
suggestion, this is kind of specific. So for when people do try to ID what bacteria are in the water, they do have primers for the 16S rRNA sequence in bacteria. So that's like something that's like it's a sequence that's specific to bacteria. Like it wouldn't pick up like a shrimp or something. Like it's only gonna pick up bacteria. And then they'll like ID, they're like slight variations of that sequence is what they use to like ID them. Um, you could do like a more general approach. I guess if you had like this fluorescent probe attached to say a 16S primer, that would pull out like a total bacterial fraction.
Yeah. Yeah, no, yeah, that's a great idea. And I mean that's just like looking at the DNA or RNA side too. Like, you could also like quantify like
Like
proteins or other
protein.
like molecules
Yeah.
as well. But yeah, like yes, the 16S, the bacterial specific like ribosome components, like that's definitely like bacteria specific and you can quantify like that like generally. I just wonder like what's the stability on that though? Because like if you have like primers, it's probably pretty unstable, especially if you're running it through like large quantities of water. And like how would you like filter the water to like find the bacteria? Like maybe that bacteria is like sedimented at the bottom, or maybe it's like floating at the top. And so
Mhm.
how do you like
Yeah.
compare the samples to find those bacteria? Yeah,
Yeah.
and I imagine this can also be applied very readily to like fresh water as well, like that you're drinking and
Mhm.
like before you
For sure.
drink it or in these other rivers as well. Yeah.
Yeah. So I guess my other questions are, yeah, the primers, that's like, I always wonder, I guess that's why I like wonder if there's, yeah, the molecular approach is tricky in that way because you have to have all the reagents, and DNA is like pretty stable, but yeah, you might, yeah, I don't really remember all the chemistry conditions that you would need to have. I think filtration would be theoretically like pretty trivial because you could just say, all right, we want to exclude everything that's like greater than some size, and then like catch all the like particles that are left. And like, yeah, you might have sediment. And so I think like the hardest part of that would be like clogging, right? If you have like other stuff or like sea foam or whatever that in there, it could clog on one of the two filters. But other than that, I think like getting the right like size group should be not hard.
Yeah. Yeah, that's great. That's a great
Yeah,
idea.
I guess a novel approach you could take this idea is in like a continuous monitoring system. I don't know if that's what you were mentioning.
Yeah.
'Cause I think definitely like this has been solved for like one time like in the lab, but having a system that can run for long durations of time, that would be neat. Yeah.
Isn't that what you're, is
I
that what
know.
No, not quite.
you're
Maybe
using
something a little different.
as well?
Yeah, like the gravity machine. Yeah.
Yeah.
Yeah, because I also read some papers where they were very fine tuned towards E. coli and the chemical, diagnostic for E. coli, and something which is continuous to actually monitor the E. coli presence in the water was something that the exciting area of research that of many, we are studying there. So yeah,
I guess a question more
because
for, oh, sorry. No, no, no.
yeah, please continue. Yeah.
No, no, this was totally separate, but like a
Yeah.
question for like Enrico and I guess like Manu later is like,
Mm.
I guess like what are like the biggest challenges right now in this area? Like what still needs to be done? Like I guess we've like hit on a couple points like such as like water quality, continuous
Yeah.
monitoring, but like are these like tractable things to work on in like a frugal science perspective? And like what are you guys maybe most excited about?
No, absolutely. I mean, like reiterating why frugal science is important. If you can diminish cost by 10, you increase accessibility by 10.
Yeah.
So like anything that you can make more frugal, it's really impactful. Yeah, current problems I would say are well, I guess last year I did a CO2 project, just monitoring like the concentration of dissolved CO2 isn't like very isn't done like frequently in the ocean, but I like your like microbial or nanoplastic approach as well. And there's lots of problems to solve. Uh, yeah. But do you have a project? And if not, let's maybe start a like a
Yeah,
thread on Discord
I
for this
think
project.
Serena, like you had a Discord, right? Or you had a
I do have a Discord group. Yeah, we have. I thought Manu at some previous point had said like a group size no larger than six. Today he said two to three. I think six should be fine, and if like that's too big, we could also like I think there's more ideas
yeah.
than people
Yeah, we
anyway,
could start
so
like
we could
Yeah.
also do two, but
small.
then becomes hard to decide which ones everyone wants to do. Yeah, so the idea that I had, I guess, yeah, Enrique, you might be tuned into this, um,
Yeah.
is a little bit different, but it is also kind of this like ocean aquatic science like context, which is like thinking about how do we like sort or isolate plankton. I'm coming at this from currently the having this challenge of thinking about how do you see like what populations are related to each other in the ocean